Hi guys, we're starting a new unit on DNA, and so we're going to talk tonight about DNA structure. And if you remember back from when we talked about macromolecules, DNA is made up of nucleotides, and you've probably heard that the overall DNA structure is a double helix. So those are the two things we're going to focus on. DNA stands for deoxyribonucleic acid. So the reason that it's so important is that it holds genetic information in all living things. And DNA is a macromolecule that's made up of nucleotides. It's contained in the nucleus of eukaryotes and the nucleoid region of prokaryotes. So things like bacteria that don't have a nucleus, they have that nucleoid region, and that's where you find the DNA. And DNA was discovered by James Watson and Francis Crick in the 1950s, but their work was built on the work of many others. Some of their colleagues were Linus Pauling, Maurice Wilkins, and Rosalind Franklin. And we'll have a chance to hear a little bit more about their story later in this unit. So let's look a little deeper at nucleotides. Nucleotides have three main parts. They have a sugar molecule, and that sugar is called deoxyribose. Remember that OS tells us it's a sugar. And it's a 5-carbon sugar, so it looks like a pentagon. Attached to that 5-carbon sugar is a phosphate group. And then attached over here is a base. And this is a nitrogenous base, meaning it has nitrogen in it. And there's 4 different bases, 4 different nitrogenous bases that can be in that spot. There's adenine, sometimes we just call it A, thymine, T, guanine, G, cytosine, C. And when DNA forms as a whole structure, you're going to have two strings of these nucleotides, and bases match up in the middle. And when the bases match up, adenine always pairs with thymine, and guanine always pairs with cytosine. So right now, I'd like you to pause this and draw and label a nucleotide with the 3 parts labeled in your notebook or in your binder on notebook paper. So now that you've drawn your nucleotide, let's look at how the nucleotides come together to form a double helix. So there's 2 lines of nucleotides that form. You see there's 1 line here and 1 line here, and the bases pair in the middle. So it ends up looking like a ladder. And it's hydrogen bonds here in the middle that join the bases. And again, you see that A, or adenine, pairs with T, thymine, and G, guanine, pairs with C, cytosine. Now, once the ladder's formed, it twists, and so it forms a double helix like this. Helix means kind of like a spiral, and since there's 2 sides to it, that makes it double. So now I'd like you to pause again and draw this in your notebook or binder on notebook paper. This is a little more in depth, right? But you don't have to put all the detail that's in this picture here. If you get the fact that the nucleotides bond together, the bases pair up in the middle, and then that it's going to be a double helix when it twists around, then that's good. You don't, like I said, it doesn't have to look exactly like this. Draw it in the way that makes sense to you. So now for standard folks, knowing the nucleotides and the double helix is what you'll be expected to understand. For honors, there's a couple extra things. So if you're standard, it's okay to stop now, or if you'd like to continue listening, that's great. For honors, this is some extra information that's expected of you. Now, when the nucleotides bond to make the 2 different sides of the double helix, the 2 sides run in different directions. You see here how this one's upside down, and this one's right side up, I guess, the phosphates. We call that anti-parallel, that the 2 strands run in opposite directions. So they're parallel, right, but they're going opposite directions, so it's anti-parallel. See that on this end, the phosphate, it ends in a phosphate here, and then over here it's ending in the sugar. Then down here it's ending in a phosphate, and down here it's ending in a sugar. We call that the 5' end when it ends in a phosphate. So here's the 5' end of this strand, and at the opposite corner is the 5' end of this strand. The 3' end is the one that ends in the deoxyribose sugar, so that's up here, and then opposite corner down here. Now, the bases, remember there's the 4 bases, A, T, C, and G. There's kind of 2 different main structures of those bases. The purines are adenine and guanine, and so if you look at this, the A's and the G's are a little bit bigger. Their structure is a little bit different than the C's and the T's, which are the pyrimidines. And I worked with a teacher once, in the way that she remembered that the pyrimidines were C and T is that cat, kind of spelled C and T, right? Cats. She thinks of cats when she thinks of pyramids, like the Sphinx is kind of like a cat. So that's a little bit weird. It might be a little bit of a stretch, but it might help you. So a pyrimidine and a purine always match with each other. So G and C match, and then A and T match. And then when G and C bond, there's a triple, 3 bonds, 3 hydrogen bonds here in the middle, whereas when A and T bond, there's just double bonds there. So that's some extra detail that we'd like you to be familiar with. I know it was a lot at once, so if you want to go back and listen to that one more time, that might be helpful.